Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied Donan membrane equilibrium and clinical applications of Donan equation. Today, moving to the second chapter by name subcellular and organelles and cell membranes. Cells contain various organized structure collectively called as cell organelles. When the cell membrane is disrupted either by mechanical means or by the membrane between the T20, the organized particles inside the cell are homogenized. There is usually carried out by 0.25 molar sucrose at pH 7.4. The organelles could then be separated by differential centrifugal forces. Albert Claude was awarded with Nobel Prize in 1947 for fractionating subcellular organelles. Here there is a diagram of cell showing different subcellular organelles. These are nucleolus, Golgi body, nuclear membrane, endoplasmic reticulum, mitochondria, microtubules, nuclear pore, lysosomes, and vacuole. Here is a table showing that which cell organelles comes out first after applying centrifugal force. Nucleus comes out first, followed by mitochondria, lysosome, Golgi bodies, and then finally microsomes. Cytoplasm you get finally in the form of supernatant containing lactate dehydrogenase. Coming to the nucleus, it is the most prominent organelle in the body containing nucleus except mature RBCs in circulation. The uppermost layer of skin also may not possess a readily identifiable nucleus. In some cells, nucleus occupies most of the available space. Example: small lymphocytes and spermatozoa. This is a diagram showing nucleus. The outermost layer is endoplasmic reticulum, and within this is the nuclear membrane and nuclear pore, and within this is the nucleoplasm, and finally there is nucleolus. The nucleus is surrounded by two membranes. The inner one is called perinuclear membrane, which contains numerous pores. The outer membrane is continuous with membrane of endoplasmic reticulum. Nucleus contains the DNA, the chemical basis of genes which governs all the functions of the cell. The very long DNA molecules are complexed with proteins to form chromatin and are further organized into chromosomes. Here you can see in a diagram that very long DNA molecules complexed with protein finally organized into chromosomes. Endoplasmic reticulum. It is a network of interconnecting membranes enclosing channels or cisternae that are continuous with outer nuclear envelope to outer plasma membrane. This is a diagram that shows uh, endoplasmic reticulum next to the nucleus. Under the electron microscope, the reticular arrangement will have railway track appearance. This will be very prominent in cells. Glycoproteins and glycoprotein are synthesized in endoplasmic reticulum. The detoxification of various structures is an important function of endoplasmic reticulum. Mitochromal cytosome, cytochrome P450 hydroxylates such as inspirin, aminopyrin, aniline, morphine, phenobarbital, etc. According to electron microscope, the ER is classified into rough and smooth varieties. The rough appearance is due to the attachment of ribosomes to cytoplasmic side of membrane, which are where the proteins are being synthesized. The cells are fractionated. The complex endoplasmic reticulum is disrupted in places and are automatically reassembled to form microsomes. Here you can see the green color as perinuclear microsomes. Golgi apparatus. The Golgi organelle is a network of latent smooth membrane vesicles. It may be considered as converging area of endoplasmic reticulum. While moving through the endoplasmic reticulum, carbohydrate groups are successively added to the nascent proteins. The glycoproteins reach the Golgi area. The glycoprotein apparatus is composed of cis, medial, and trans cisternae. The glycoproteins are generally transported from endoplasmic reticulum or to cis, cis Golgi or proximal cisternae, then to the medial Golgi or intermediate cisternae, and finally to trans Golgi or distal cisternae for temporary storage. Trans Golgi are particularly abundant with vesicles containing glycoprotein. Newly synthesized proteins are sorted out first according to the sorting signals available in proteins. Then they are packaged into transport vesicles having different types of coat proteins. Finally, they are transported into various destinations, which is energy dependent process. Main function of Golgi apparatus is protein sorting, packaging and secretion. The finished products may have any one of the following destination. First, they may pass through different membrane to the surrounding medium. This forms continuous secretion. Example, secretion of immunoglobulins by plasma cells. Second, they reach plasma membrane and form an integral part of it, but not secreted. Third, they are formed into a secretory vesicle where these products are stored for a longer time. Under appropriate stimuli, the contents are secreted. Release of trypsinogen by pancreatic acinar cells and release of insulin by beta cells of Langerhans are cited as examples. Fourth, the synthesized material may be collected into lysosome packets. Here comes the end of today's session. Please like, share and subscribe the channel. Thank you.